patients with metastatic breast cancer, an understanding of tumour genetics will drive new treatments. There are many different types of breast cancer, and this might be driven by changes in genes or proteins, and those changes themselves are targets for drugs. The more we understand about the genetic changes in cancer, the better able we are to target different changes and create better drugs. A so-called personalised treatment.
A small minority of breast cancer patients are carriers of one of the germline breast cancer susceptibility genes, BRCA1 or BRCA2. Part of the pathogenesis of cancer in these patients is an inability to repair DNA damage. And the cancers which they get can be a little bit different.
The main option you're trying to give them is to give them a DNA repair related therapy, okay? Because we know that in these patients who have these mutated genes, the mechanism of DNA repair is not working.
The PARP inhibitors exploit the weakness, and therefore the cells are pushed into apoptosis or programmed cell death.
Talazoparib is a very special molecule, and we know from preclinical studies from tumor cell lines, but also from the animal model, and now also from the human model in phase 1 and 2 studies, that this molecule is the most potent inhibitor of the PARP molecule. We know from the drug that it's acting very specific, and it's inhibiting those BRCA1 and 2 mutated cancer cells. So therefore, we are quite excited that we can offer our patients with BRCA mutations the new opportunity to be treated with this drug within the framework of a worldwide study.
And that is really where we are today with this very exciting trial, the BRCA trial, which is going to look at talazoparib specifically in patients that have a BRCA mutation.